Let's move on to the cytoskeleton. Do you remember the cytoskeletal elements? We've got microtubules, actin, and intermediate filaments. We call it the cytoskeleton, but it's not just to hold the structure of the cell. It also has roles in transporting things around the cell along microtubules, helping the cell move around using flagella, and even helping the whole body move, since muscle contraction depends on actin and myosin. You can get a sense of how the cytoskeleton is organized by this picture, where the nuclei are blue, the microtubules are green, and actin is red. Actin and myosin compose microfilaments and are the thinnest structure in the cytoskeleton, with a diameter of just 7 nanometers. You probably know that actin and myosin work together in muscle contraction, and that's covered in a different lecture. But can you think of anything else that actin does? It's an important structural component in microvilli. Do you remember where you might find lots of microvilli? One example is the brush border of the small intestine, where microvilli are used to increase surface area and therefore increase absorption. Remember, a villus has a lot of cells, and each cell has lots of microvilli, so microvilli are a lot smaller than a villus. Actin is also used in adherens junctions, which are common in epithelial and endothelial cells, and are used to hold cells together. Intermediate filaments are generally involved in cell structure and have an intermediate diameter of about 10 nanometers. Some examples of intermediate filaments are vimentin, desmin, cytokeratin, glial fibrillary acid proteins, and neurofilaments, but I'll come back to these in a couple of minutes. And microtubules are the largest part of the cytoskeleton, with a diameter of about 25 nanometers, and they're generally involved in movement of things within the cell, or of the cells themselves. We see them in cilia and flagella, and also in the mitotic spindle and centrioles. Okay, sorry for jumping around a bit, but we're going to revisit intermediate filaments in the context of histological stains. Glial fibrillary acid protein, or GFAP, is found in neuroglia, such as astrocytes. Neurofilaments are obviously found in neurons, and cytokeratin is found in epithelial cells, which you can remember since epithelial cells in the skin are keratinized. Lastly, desmin is found in muscle tissue, and vimentin is found in connective tissue. There's not much else to say about these, just make sure you know which stain is for which cell type, so when it shows up in your exam it'll be that much easier to answer the question, or at least rule a few answer choices out. Okay, let's talk more about microtubules. What color are they in this image? They're the green ones. They're about 24 nanometers in diameter, which makes them the largest cytoskeletal element. They're cylindrical structures formed by polymerized dimers of alpha and beta tubulin. Each dimer has 2 GTP bound to it. Also remember that they have a positive and a negative end. They grow together in chains, maintaining their polarity. While their growth is relatively slow, they can collapse quickly when needed. So what are microtubules used for? Well, there's really a lot of things. Basically, anytime you want to move something large around a cell, you need to use microtubules. For example, during cell division, what do you want to move around? This would be chromosomes, since you have to separate homologous chromosomes so each cell ends up with a copy. So microtubules, which are these green lines, are anchored to centromeres and pull them apart. What else can we move around? How about carrying vesicles filled with proteins along an axon of a neuron? Now, as I mentioned before, there's a positive and a negative end, and you need a different motor protein depending which direction you're going. If you want to carry something towards the positive end, which is called anterograde transport, you can attach your cargo to a motor protein called kinesin. And if you're going towards the negative end, which is retrograde transport, you can use the motor protein dynein. In neurons, the positive end of the microtubules is at the distal end of the axon, whereas the minus end is near the cell body. So which motor protein is used to move something towards the synapse? That would be kinesin. And by the way, if you're moving a vesicle filled with proteins along the axon using kinesin, where were these proteins synthesized? If they're in a vesicle, then they were made in the rough endoplasmic reticulum, not in a free ribosome. And remember, in neurons, the rough ER is called the Nissl body. The last function of microtubules I want to cover is their role in cilia and flagella, but I'll save that for the next slide. First, let's go over some things that interfere with microtubules. Alright, so why would you want to interfere with microtubules? Well, like I said earlier, one of their functions is in cell division. So anytime you want to block cell division, you can use a microtubule inhibitor. Some examples of microtubule inhibitors are drugs that treat cancer, gout, and parasitic infections. Cancer should be obvious here since cancer cells divide a lot, but since immune system cells also require lots of cell division, microtubule inhibitors have kind of an immune suppressing effect too, which is why they can treat gout. And for the parasites and fungi, you want to use inhibitors that are specific to those types of species and have minimal effect on human microtubules. Some examples that are most likely to show up on step 1 are mebendazole, which treats helminths, cruciofulvin, which treats fungi, colchicine, which is a super important drug which treats gout, and vincristine, vinblastine, and paclitaxel, which are all chemotherapy drugs. Now, why does colchicine treat gout? Well, first of all, what is gout? Basically, if you have high levels of uric acid in your blood, it will crystallize. And these uric acid crystals will deposit in joints, especially the big toe. This ultimately causes recurrent acute inflammation. Alright, so what drug can you use to treat patients long-term for gout? That would be allopurinol, because allopurinol prevents uric acid formation. But during an acute attack, what do you use? So that would be colchicine, which doesn't affect uric acid directly, but is basically working as an anti-inflammatory drug, and you could also use NSAIDs or ice. Now for these chemo drugs, you learn more about these in the Hem/Onc chapter, but what do you think might be an important adverse effect of inhibiting microtubules with vincristine, vinblastine, or paclitaxel? These drugs can cause bone marrow suppression, since again we're preventing cell division. And like it says in your book, you can remember these with a mnemonic: microtubules get constructed very poorly. Alright, so what happens if microtubules aren't working properly? Well, it depends on the specific problem, but one example is called Chidia-Higashi syndrome. This is usually caused by a mutation in the lysosomal trafficking regulator gene, or LYST, which normally is required for organizing proteins into mature endosomes for microtubule-dependent transport. Since granulocytes need this to make their granules, this defect makes patients susceptible to recurrent pyogenic infections, since macrophages and neutrophils can't make the weapons they need to destroy bacteria. These patients also have albinism, since melanin can't be transported out of melanocytes, and peripheral neuropathy, since vital proteins can't be transported to the ends of long peripheral neurons. So as I mentioned a minute ago, another role of microtubules is in cilia and flagella. Both cilia and flagella are made from microtubules arranged in a 9 dimers plus 2 singlets formation. So here I'm showing you an electron micrograph of a cross-section through cilia, and you can see the 9 dimers around the periphery and the 2 singlets in the middle. Now in the last slide I mentioned that dynein is used for retrograde transport along microtubules. Dynein can also be used to bind microtubules for a different purpose, which is to link these doublets together. Then ATP can be used to cause them to slide along each other, which makes the cilium bend. Flagella and cilia have a similar structure if you take a look at the cross-section, but they also have some functional differences. First of all, flagella move in this kind of propeller pattern, since they're trying to generate force that pushes the cell away from where it's pointed. Cilia beat back and forth, since they're trying to keep the fluid outside the cell moving in a consistent direction. Flagella are a lot more common in single-celled organisms like bacteria, but can you think of any human cells that have flagella? Sperm are the first ones that come to mind for me. How about cells that have cilia? Two important ones are cells in the lung that keep mucus moving out of the lungs, and cells in the fallopian tubes, which move the egg towards the uterus. Okay, so what happens if you have an autosomal recessive defect in dynein? This is called Cartogner syndrome, or primary ciliary dyskinesia, and if you have it, your cilia and flagella won't be able to move at all. So all these functions I just mentioned won't work. Both males and females will be infertile, since sperm can't move and the egg won't be pushed along the fallopian tube to the uterus. Patients with Cartogeners also commonly have bronchiectasis and recurrent sinusitis, since cilia are required to sweep particles out of the lungs. And lastly, it's associated with situs inversus, which is a condition in which major organs are found on the opposite side of the body. For example, the liver should be found on the right side, but here it's found on the left. Before we move on, let's see if you remember which direction these proteins go. Dynein is used for blank transport, and kinesin is used for blank transport. The options here, by the way, are either anterograde or retrograde. The answer is retrograde, or towards the minus end for dynein, and anterograde, or towards the positive end for kinesin. The plasma membrane separates the inside of the cell from the outside, and also is responsible for mediating interactions between cells. As you can see in this image, it's asymmetric, so one side may not look like the other. It's mostly composed of an even mixture of cholesterol and phospholipids, but also contains some sphingolipids, glycolipids, and proteins. Some proteins, like this one or this one, are transmembrane proteins, which means they have an intracellular, transmembrane, and extracellular part. These include transport proteins and ion channels, and receptor tyrosine kinases. There are also peripheral membrane proteins, such as phospholipase C, which are anchored to the membrane either by binding to another protein or a covalent bond to a membrane lipid. It's important to know that hydrophobic domains of proteins will always be found in the transmembrane region, since they're repelled by the water that is outside or inside the cell. Hydrophilic domains will be found bordering the cytosol, extracellular domain, or within the lumen of a transport protein. The sodium-potassium ATPase pump is a transmembrane protein that helps maintain physiologic concentrations of sodium and potassium in the cell. To do this, it utilizes active transport, which means it hydrolyzes ATP. Since ATP is produced inside the cell, it makes sense that the ATPase domain is on the cytosolic side of the protein. Remember, the inside of the cell has a lot of potassium and only a little bit of sodium, whereas the outside of the cell has a lot of sodium and only a little bit of potassium. Therefore, the role of the sodium-potassium pump is to bring potassium in and sodium out. For each ATP used, 3 sodiums are transported out of the cell and 2 potassiums are transported in. So let's say you have a cell and somehow shut down its sodium-potassium pump. Well, what's going to happen? First of all, sodium will start to accumulate inside the cell, since you're not pumping it out. And whenever sodium concentration increases anywhere, water is going to follow it. So the cell will swell and eventually lyse from osmotic stress. So this pump is really important in all cells, but there are some cell types that make their sodium-potassium pumps work really hard. Can you think of an example? One would be neurons, since every time an axon potential goes down a cell, they need to repolarize the cell. And a second one would be muscle, since even though they're dependent on calcium concentration for contraction, the sodium-calcium translocator uses the sodium gradient to remove calcium from the intracellular space, since sodium wants to flow into the cell, and you can use that energy to push calcium out. The sodium-calcium exchanger, by the way, does not use ATP. It just operates through passive transport. There are a few drugs that inhibit this pump. Wabane does this by binding to the potassium binding site and is used as a poison by hunters in Somalia. Cardiac glycosides are drugs used for arrhythmias and congestive heart failure, and they include digoxin and digitoxin. These directly inhibit the ATPase part of the pump, which then causes sodium to build up inside the cell, which in turn causes the sodium-calcium exchanger to allow sodium to leave the cell. This ultimately increases the amount of calcium inside the cell. More calcium in cardiomyocytes means more cardiac contractility. Alright, time for another flash quiz. Let's see if you can remember those immunohistochemical stains for intermediate filaments. So what cell type is stained by desmin? And the answer to that one would be muscle.